Let me play for you a little bit so you can hear the difference. Let's see if you can hear the difference. So I will play the birdsong song and then this stimulus. So it's a repetition of 3 times, but it's just copy-paste. Okay, it's the same, but I just did that because this is how the zebra finches sing. It's not just one motif, they repeat it. So if I increase the noise, this is kind of not so loud, and in a noisy environment, you cannot tell the difference, right? So you can see it here. So for example, here you can see that this is no noise, and this is more noisy. Okay, in the spectrum you can see it, but we as humans, we cannot hear this too much. And now when I increase the dissipation, I think you would be able to hear it. Okay, you can hear more that is interesting because you cannot see it so much in the spectrogram, but in the sound it's very obvious. So now with all these stimulus, we test the neurons and say it doesn't matter now what we hear or what we quantify, we want to see what the neurons are saying. And these are the results. So when we have no noise, so again in this thing, when you have no noise, you can have a little response or not response at all. Then if you increase it, you have more response, and then you increase it, you start decreasing the response. But whenever you increase the dissipation, so when you start removing this sort of pharyngeal cavity, these Helmholtz resonators, the neurons say, no way. Okay, so it looks that it's much more important, the resonator, than the noise. No noise is important. They don't like it. But not having the appropriate filters, that kills it. Okay, they say, no way, this is me. And actually, we keep on increasing the noise. And what happens is that you just get to a plateau. Yeah, I will show that later. So what we gain information here is that indeed these are relevant parameters, and now we did so the noise and the dissipation are important and physiological parameters. It's not that you just got lucky once, okay? If you're tuning the parameters and you see differences in the neural response, it's because these are key parameters for the physiology, for the biomechanics, for the birth control. These are group data, so we just choose the zeta score, and we are just, it's a way of quantifying the amount of response you have, and we are dividing by the Bose response. So a perfect response will be of 1. Our birth response was 0.6, let's say. So still, and you can see it here, for example. So this is our best shot, okay? Some noise and with the resonator. These are the same scale. So what you can see is that if this is the birth response, this is responding in the same point, and this is hard to get, but the probability of response is not the same, okay? So, and what is interesting is that if you look at the raster, you can see that either they respond or not, but you have less probability of responding. You have gaps here, but when you are presenting the birdsong song, they respond every time, okay? For the best synthetic copy, there are some places where still the model is not so good enough. But it's good enough for us to ask these questions, because whenever it's responding, it's sharply responding in the same place. So this is, and so whenever you have the noise, if you increase the noise, so whenever you have no noise, you have no response, and then when you increase the noise, again the response decreases, but it goes down to almost zero if you start removing the oropharyngeal cavity, okay? And this is interesting because this resonant cavity, what it's saying is that it's a relevant feature, and actually it's a thing that they can control when they are singing. In the same way we can change the volume of our mouth, the birds can do the same with the volume of this resonator. And actually it's been shown that they use it for tracking the fundamental frequency and removing the harmonics, and in the zebra finches to emphasize this region. And the noise, here is the expansion to other places because, okay, it's almost zeros, it's the best match, and then decreases, but then doesn't go to zero anymore. And these are really, really noisy. These are really noisy stimulus, but it looks like these neurons are tuned to handle noisy environments. And actually zebra finches in the wild, they are like in huge communities, so it's a noisy environment, nature is environment, so it looks like no noise is no physiological, but heavy noise, we don't care, okay? You need some noise in order to be, well, you are not a robot, you are something natural, okay? But there are some noise that maximizes the neural responses. So this is one thing you can do with these static parameters, okay? Because then you will say, well, our best fit is going to be this, with a resonator and with some noise, okay? But then, still with this, my boss was not happy. He was saying, okay, I don't care. Okay, you discovered that the two things that you added, you still are telling me that you are a good modeler. And I was telling him, I want to go home. I'm done with it. I don't want to do any more experiments, okay? I started this to do electrophysiological experiments, and then I wanted and say, I want to stop. That's it. Enough for me. Because, I mean, these were like so many stimulus, and this was it. These experiments were done in sleeping birds. So these selective properties emerges when zebra finches are sleeping. If you play back when they are awake, the neurons don't respond. So these were experiments where you do a craniotomy, you fix the head of the bird, you do a craniotomy, and then you go like fishing neurons. And these were very long experiments. And yeah, many months with each bird. But what he said is, well, let's try. So we have like the motor gestures, right? So we have what the tension looks like and how the pressure looks like and what is, so these are much simple features than if you look at the whole complexity, okay? So this has like an infinite complexity if you look at it. Or very high, at least. But this is just like the backbones of your behavior. So his suggestions was, why don't we start looking where the neuron fires, but looking at this, not looking at the sonogram. And we did that, and we started to see that, so these are the rasters, and these are sleeping birds, as I told you. And so it looks like the neurons didn't fire in any place. It were firing in these specific places that we call GTEs, or gesture trajectory extrema, because it were at the end or at the beginning of the gestures, or also in the peaks. Let me show you another example. Okay. So this was done in sleeping birds, and I have other examples, doesn't matter. And so our claim was these neurons are not firing everywhere, they are firing in these specific places related with motor gestures in sleeping birds. So then then you have to do experiments in singing birds, because if you are going to talk about motor control, doesn't count if you are doing this in sleeping birds. We were doing in sleeping birds because the pattern of response is the same as when they are singing as when they are sleeping, but still, if you want to publish that. So I had to go to Zurich to learn a new method to record from singing birds. Went back to Chicago, do the recording in singing birds, and we send it to nature with this bird as an example. Look, in singing birds works in the same way. So they are firing whenever, so this is the raw data, and you can see that this is firing at the end or in specific places that, well, here you cannot see very well. But for example, here, it's just in the, when one gesture finishes and the other one starts, just in the transitions of gestures, okay? So this was my first bird. When you go and try a technique, sometimes if you're lucky, this works for the first time, it works. We send it to nature and they say, well, this is interesting, but now go ahead and do more birds, because with one bird, we cannot do anything. I was so ready to go home. So I had like 3 months before my fellowship ended. And I was done already, okay? And in these 3 months, I have to do all the other recordings. And obviously, it didn't work. So when I said, well, this works, this is so easy, and then you start doing experiments with all the pressure that you just have a couple of months so that nature will publish your paper, nothing works. But at the end, it worked. I was recording until the day I took the plane, practically. Not that, but I left the bird implanted, for example. So another student will continue with the work. And so there's some results. So this is a raster, and you can see that, so the neurons fire, so different colors are different neurons. They fire at the end, at the beginning, and then in some places that it's not so obvious. So this is kind of obvious, but there are some other places that they are not so obvious. But if you look at so in the sonogram it's not so obvious, but if you look at this gesture, it is obvious because it's when you, this gesture changes. And what is interesting is that with singing birds, we model, we can model before or after, doesn't matter. But when we discovered this, these were sleeping birds, and the same thing happens. And the modeling was done before our analysis. It was not that we were doing the analysis and changing the parameters so that it will fit our idea. This was done, this interpretation was done afterwards, okay? That is not a good argument for a journal. Nobody will buy it because it's just a leap of faith. I'm swearing to God, I did it afterwards. Doesn't work like this, but to us, it was very important because we really believe it, okay? Okay, and this is why this paper went to Nature. Actually, for us, physicists, the more important result of this paper is the modeling. And actually, Phil Holmes, when he cited this reference, is the only other one that gets it. To us, this is important. But this opens a huge debate. So this was also done in the same journal that when we published this, it was a comment showing why this was important. Because before us, so this goes like this. So this is modified because the one that made the cartoon didn't work, did it right. So I modified it. But the idea is you have a song, so special notes, and you have recorded activities that are so where you cannot record all the neurons, right? So you just can record a few. Doesn't matter if it's 10 neurons or if it's 100. It's a few compared to the 100,000 of neurons that you have in this particular nuclei. So you record a few, and you want to see how is the coding in relation with the song. And you have two hypotheses. that this is a sequence, so it's the clock hypothesis. So you are recording a few, but actually you have an underlying sequence, or you have the gesture transition hypothesis, that is, you don't have a sequence, you have gaps, okay? Neurons are not coding for all the times, they are just coding to specific times, just saying, well, this is an important place, and what is this, and this is what we think. And we think this happens because the behavior emerges, again, from a whole brain interacting with the body and in a complex neural network, okay? So in this place where we are recording, that it's one specific place in the brain, you just need places that this part is important, this like a kicking, so here, 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 and then the whole neural network gives them more complexity and the body gives them more complexity, okay? So we started this debate. So these guys, the clock hypothesis guys, Michael Fee started it, but then, so he's an engineer, and it's a very linear way of thinking, but yet he's very intelligent. So we get to have very interesting discussions with Michael Fee. Michael Long, his PhD, it's not so good to discuss with. The problem is that much of his research, he's in the United States, and he depends on grants in order to make his living and have power, okay? So there's all this sociology, if you have more grants, you have more power and all that. And so I don't know if he believes it or that he wants to be true and he doesn't want to lose his power, but Michael Long, but in any case, these are very powerful guys. And we are from Argentina, South America, and with Dan Margoliouth, that it's not so powerful right now. And then I discovered that the problem was that Dan Margoliouth and Michael Fee had this huge fight years ago. So I was there with my result, but actually this was because they were still fighting because of an intellectual fight they had 20 years ago, as always, right? So we are people doing science, and so, okay, doesn't matter. So the problem is, okay, we have these two hypotheses. Now, we want to, if you are like, if you still believe in science, you want to know how the brain is working, okay? So, we started to look at this in detail and say, well, the problem with the gesture transition hypothesis is that you have to model and you have to extract these complex gestures. But at the end, the only thing that matters is where are the transitions, okay? So we develop a method to extract these transitions directly from the sonogram, okay? So we did that for zebra finch, and what we started to notice, this is a zebra finch song, is that zebra finches have very complex songs. So each line is a GTE, is a gesture. So if you look at this, and if you just organize it like in GTE, and so these are so complex that this looks like a sequence, okay? So for you to understand, the GTEs should look like this, with gaps. But if you look at the GTEs in the zebra finches, it looks like a sequence, okay? So this is the GTE models, but look like a sequence. So the result is that GTEs, you cannot disambiguate these two models using zebra finches, because zebra finches, let me say it in a different way. These bursts last around 10 milliseconds. And you can find gestures that are 5 milliseconds apart, 10 milliseconds apart. So the whole sequence, so the whole birds are overlapping, okay? They look like this. If you would record one neuron per gesture, you will see this, okay? published this in 1919, nobody cared about it. They are still recording in zebra finches because zebra finches are tough. They are good animals to breed, so people like zebra finches. And we like it too, but these are not a good animal model to disambiguate between these two models. Okay, not the most suitable zone to disambiguate between hypotheses. So we started to look to canaries. So canaries has a different zone. We did the same thing. So the difference with canaries is that you have a syllable and they repeat it, another syllable and they repeat it. If you do the same thing of GTE extraction, you can see that zebra finch, it doesn't matter how you do it, it's always a line. But with canaries, they start like going apart from a line. Moreover, in order to compare zebra finch with canaries, the problem is that they have like different sequencing thing, right? So we built what we call a canary pseudo motif. That is, we take one syllable, no, because in the motifs of the zebra finch, you have like A, B, C, D. And in canaries, you have A, B, 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 C, 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 C. But in order to compare, we get one syllable of each canary, of each phrase. We get one syllable of phrase 1, one syllable of phrase 2, etc. And we construct this. So here you can see the different syllables, and you can compare more directly. So different A, B, C, D, or 1, 2, 3, 4. Okay? Yeah.
So I think I didn't understand why zebra finch is not good, but canary is beautiful.
Yeah, because canaries have gaps. Canaries have these syllables that are like harmonic stacks, but that last like 200 milliseconds. So the prediction of our model is that in these 200 milliseconds or 60 milliseconds, you wouldn't expect to have any neural activity of projecting neurons in HVC. So it's because of the gaps that if you say, well, if this is coded as a GTE model, you would expect to have neurons recording here, but whenever these syllables comes, you would expect to have one recording here, and nothing here, and another one here. From phasic projecting neurons. Then you have interneurons. It's not that the brain is shutting off, okay? So whenever you see nothing in the projecting neurons that are excitatory neurons, it's because you have inhibitory neurons that are firing like crazy, okay? Yeah. And the thing is that if you do the same thing for zebra finches, like each line, if in each line you find a burst, then your burst will align with no gaps, almost with no gaps. Actually, if you look at Michael Fee or Michael Long papers, and you look at long harmonic stacks, you will see that they don't find bursting. Actually, it's already in their data. The thing is that they kind of cheat to say, well, there is not such a gap, I don't know what, but it's already there. So whenever you see like this, you expect to have no bursting, okay? Yeah, so it's like this. So with canary pseudomotifs, you would expect to find, for example, one bursting here, so one broad dot here. No, these transitions are like these transitions. So you would expect to have, for example, this, and you will find a gap here, that it's a gap here, if you record all the neurons. And with zebra finch, no, okay? So that is why we started recording in canaries that are much harder to record from than, and they also just sing during spring and summer, so no summer vacation for the students and for myself in the first years, okay? So it's tough. Yeah, so these are the people, before I forget, and so these are my collaborators, Gabriel Mindlin, with I share the lab with, and Dan Margoliash, the students that took part of the whole project and the founding agencies. Now, I wanted to tell you a little bit about the Canary recordings. How much time do I have? Just like 1 hour. Okay. Sorry, before moving on, do you have any other questions regarding the other? Because I will tell you a couple of things. No, yeah.
Just what kind of algorithm do you use to fit the model to the birdsong?
Yeah, so this is what Daniel was calling yesterday brute force. So we do an algorithm. What we did was just we fixed. Let me put wait. Share. Yeah. So, so we have two parameters, alpha and beta, or pressure and tension, and we fit these parameters so that the sound that is produced match the same fundamental frequency and the same spectral content. That was the algorithm, okay? So it was just building a grid of values of alpha and beta and fundamental frequency, a grid of alpha and beta and spectral content, and then match it. The other constraint is that we wanted we don't want it like jumping, so things of continuities, unless you see a discontinuity in the sound, something like that. But this is brute force and something done by hand. That is why also we wanted to do another more automatic algorithm for GTE's extraction, because this was a lot of by hand and it was a lot of work and you need to know about modeling. But this other tool of extracting the GTEs by the sound, just that now it's more automatic. But again, you have some constraints. The constraints there are some features that you extract from the sound envelope mainly. Okay, great. Yeah. So actually, Felipe, when he started the PhD, he said, come on, really by hand? What are you talking about? And so, and I said, well, if you want to do something else, be my guest, but I want you to model Ornero songs, not in a year, but in a couple of months, because we want to publish a paper. So he's now struggling to say, what do? I do what I think I have to do, or do I do what my advisor is telling me? We don't know. I just want the results. Whatever the best results, I mean, if you have an algorithm and an automatic way, and it's better for everyone, because then other people can reproduce your data. That is what we want in the scientific world, okay? It's not just me, I'm the god of modeling, and you have to ask me and kiss my ring so that I model for you. Thing that we don't like to do, because we want to model to ask our own questions. But yeah, so that's the struggle. Yeah, sorry.
Data, it's an open database, or you're collecting in a laboratory or in the nature? How do you search this data?
I don't understand the question. So yeah, the data is available, and if you ask me, I can send it to you. Back then, so this is a 2013 paper, it was not so many regulations about the data, but if you ask me, I can send.
Like the bird songs, the algorithm of the bird songs.
The what?
The
So,
algorithm
Why don't you ask me in Portuguese?
Qual que é a fonte do seu dado? Tipo, como você coleta? Onde é a gravação? Você usa a gravação no laboratório, gravação na natureza? Tipo, da onde está vindo essa informação? É um banco de dados aberto?
A gravação do áudio ou de...
Exato.
Okay, so so the data collection, this was done in the lab. The audio is a microphone, and then so it's just a microphone in sound boxes that isolated sound boxes, and those are good microphones. So it can be directional microphones, so you have like a very clean recording. And then we have neural recordings, but the recordings, and this is why this looks so nice. If you're in nature, everything is much more noisy. But still, depending on this is why if you're in nature, I will recommend you to record from more tonal birds, because tonal is more like a very defined sound, and you can separate it from the background noise. If you want to record a noisy bird in the nature, good luck. It's going to be hard for you. Other questions? Because I will now tell you a little bit of how we continue the story working with canaries and what we find, but it's like a different thing. Okay? And if you have more questions regarding this or regarding the modeling or regarding how we get to the model or whatever, just ask me later, okay? Today probably I will be tired, but tomorrow I will be fresh and happy to talk. So, let me see. So here, so here is what I was telling you. That maybe you can choose what you want to hear in the last story. So one story is we have this thing that we are saying the brain is coding in these particular gestures. So what you will find in the brain is related to what the bird is singing. So we made several recordings in anesthetized canaries and in singing canaries and all that. So I can tell you about that part. Or I can also tell you about, well, singing canaries, again with the same question. We record it from some neurons and we make a model of the neural activity. And in that way, we kind of say, well, with these recordings and this model, we show how this could work in the brain. Okay? So it's all different stories following, we trying to show the world that how is this part of the brain coding for behavior, but one involves modeling and the other one don't. So who votes for no modeling, just data? Okay, who votes for modeling? No, you have to vote. Okay, so modeling, okay. I can tell you about the data. I can tell you about all the other part later. Let me, why do you do that? Okay, let me just show you a couple of slides. Okay, so let me, in any case, introduce you to the data and then we'll do the modeling. So the raw data is like this. So we put an electrode and we do extracellular recordings, okay? So these are extracellular recordings. No, neurons. Neural extracellular recordings. So we put an electrode that it's a metal electrode, and we are going to record some neurons all around. So this is the raw data, but then you can do a low pass filter, and you will have what we call local field potential, so these like slower oscillations. Or you can do the high pass filter and just get the spikes. Okay, and depending on the amplitude, you can get different neurons. So this neuron is the same, and it's very close to the electrode, and these small spikes are from neurons that are kind of far away from that. Okay? So the cool thing about this is that we can just keep the spikes, or we can put a threshold and just count spikes and get a profile of multi-unit activity. So wherever you see a peak in these MUA profiles, is because you have a lot of spikes happening simultaneously, okay? So you can choose either keep the spikes or say, well, I'm not going to do hand picking, I will just get all the spikes and just sum the spikes and do a sliding window, and you have that, okay? That is one piece of information that is important. Okay, any doubts? Okay, these are sleeping, whatever. As we are in Argentina, and we know economic crises are coming, if we are not in an economic crisis now, it's because it's coming in a few years. It's like that in Argentina. If you don't plan for an economic crisis, the problem is you, because you know the country already, okay? That's the thing. So, Daniel knows what I'm talking about, because Brazil is much robust, but crazy politicians are all over the world, okay? And they suddenly decide that science don't care, and then suddenly decide that science is the best thing ever. So, as we work with animals, so our approaches are either go to the field and do great things in nature, because you are in a privileged place in nature that we are in Latin America and that has privileged environments and species and great behavior, so you can either do that we are doing this, or you can get some, build your own electrodes, for example. So this is another approach that gets you to build tetrodes. The advantage of this, so this is a 16 channel, so these are the tetrodes, we build it. So this is just the connector and the micro drive, so you screw, whenever you screw and unscrew, you can move the electrodes up and down, and you can choose as many channels you want, depending on your amplifier. We started with 16, and the advantage of this is, as you are building it, you can put the geometry that you like, because you are building it, okay? Let me show you some data. So this is a canary singing, and here are the 16 channels. Each row is one tetrode. So as you see, the tetrode sees neurons that are very similar. The advantage of measuring with a tetrode is that if the neuron is here and the tetrodes are here, as you have different. No. So do you know about tetrodes, or shall I talk a little bit about it? So tetrodes, it's something that is widely used for poor people now. So you have the 4 electrodes, and you have the neuron. right? It looks like a flower, but it's, I don't know, the neuron. So what's the idea is that the distances from the neuron are different. Okay, you have like L1, L2, L3, L4. The distances from the electrode to the neuron. And as I showed you before, if the distance are different, you will have different amplitudes of the spike. Okay, you will get this, the 4 electrodes will get the same spike from the neuron, but they will see it. For example, this one, that is L1, will see a bigger spike, and this one, that is L2, will see the same spike, but smaller. This would be tetrode 1, this would be tetrode 2, okay? And this is how you can separate the spikes. This is why we work with tetrodes. Okay, so what you are going to see is that in each, but you see almost the same in one tetrode, but if you are a tetrode that is kind of more far away, for example, this and this, you can have, you see different spikes, okay? So this here is the bird singing, and here we are choosing which channel to see. So here you will see the spike. Another piece of information, as this is voltage over time, if you plugged it to a speaker, each spike sounds like a click, okay? So whenever you see a click, it's because it's a spike. Here, that you have a lot of spikes, you will see like, okay, that's what you hear. It's a lot of neurons firing. Are not firing. And the people that it's saying that this is not coding for behavior haven't heard that already, okay? Because it's so obvious. The bird sings like beep, 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 and the neurons are like So I will play back again this for you. Okay, pretty obvious. I rest my case. So, if you look at LFPs, local field potential, in singing canaries, it happens that the syllabic rate and the LFP rate match. So this is an identity curve. And this also happens for anesthetized birds, okay? So the rhythm in the behavior match the rhythm in the brain. Okay, that's the take-home message. And you can look at the rhythm, looking at the oscillations in the local field potential, or you can look at spikes. Okay, but the problem with LFPs is there is always a debate that what is LFPs? What are LFPs saying, measuring in the brain? Is the activity of multi-unit activity, or is the input? Okay, so that is a question and it's a debate, and we can talk about it later with the data, but you wanted to hear about modeling. So to us also this goes in the direction that this part of the brain is coding for behavior. It's not a sequence, it's related with behavior, okay? Because if you see a rhythm and the behavior has a rhythm, then it's obvious. The problem is that zebra finches doesn't have rhythm, doesn't have an obvious rhythm. And that's also the approach of neuroethology. Whenever you want to study, choose the species that it's the champion of what you want to understand. And that is much obvious. And that is how science moves so forward. Look at, you have 4,000 species that learn how to vocalize and that they have complex vocalizations. Choose the species. Don't stay with working with zebra finches that are the guys that are easy to work in the lab. And that is so what we are doing, cannabis. La, la, la. So, now the other approach is just keep the spikes. And again, for example, you see each spike corresponding to each note. And for example, here, these long whistles, you see spikes like here. But again, the problem is you're just recording some neurons, there are few neurons, but you need a model to fill in the blanks if you want. Okay, so if you record neurons, you will always record from few neurons. So modeling will give you a tool to say, well, with these neurons, can I make this work? How would it work in the brain? So we build a neural model that can generate complex behavior, motor patterns when singing with minimum ingredients. So now you have to look at the brain. So these are the neural nuclei. So again, these are dinosaurs, birds, don't have layers and all that, like neocortex don't have. But they have structures that are called neural nuclei that are very similar in a structured way as mammals. So you can talk about cortex, or you can talk about thalamocortical loops and all that. But here you have different neural nuclei that are in charge of different things. So you have the auditory input. Then in pink, you have neural nuclei that are engaged in the learning process. And in black, you have the motor part. So we're just going to remove everything else and just keep the motor part. That is also interesting because you have also this loop, right? So you have an input, or doesn't matter, but then you have UVA, HVC area. This goes to the motor output, but then you have a feedback loop. So if we just model this, we say, well, DM could be this, what we call initiating area. Then UVA, then HVC, and then array, and then to the behavior, to the muscles. So these are respiratory muscles, these are expiratory muscles. But also you have DM goes directly. So you have one loop that goes from the, this is the thalamus, and goes to the cortex, and goes back to the behavior, or you can go directly to the behavior, okay? This is the neural anatomy. And this is like the minimum thing. So what we did, like modeling, okay. And then inside, so this is one neuron that it's called array, and you have excitatory and inhibitory. In HVC, you also have excitatory and inhibitory. We just make this population of excitatory and inhibitory neurons for some of them. And again, this is plain GAL. So this is like the simplest model ever. So this is just the excitatory and inhibitory populations. And this is just that. So this is a sigmoidal function. I think Daniel was telling you about this model yesterday or something like this. Doesn't matter. So you have, this is just an exponential decay, and then you have constant inputs, or you can have also in this sigmoidal the excitatory input, like making like auto-excitatory, and it's a minus because the inhibitory reduce the activity, okay? And the same with the and they are interconnected, okay? And again, here, if you have like, you're recording from several neurons, whenever, this is like a MUA profile, okay? Whenever you see a peak is because you have many neurons firing at the same time, okay? So this is what we are going to have as an output for the model, okay? And so this, for example, excitatory inhibitory is just for some of them. And then if not, they are just excitatory neurons. Yes?
This is a neural mass model.
Sorry?
Is this just like a neural mass model?
Yeah, so it is called additive model or, yeah. Yeah.
It's not like a...
We are not coding.
It's not cell-wise.
Yeah, we are not coding for the spikes.
Oh, okay.
Yeah, we are not coding for the spikes. We are just coding for we are not coding for these spikes, we are coding for this. Okay? So so it would be like you are recording from a multi-unit activity. So instead of getting the spikes, you just get the multi-unit profile. And that is what you will have. Yeah. That allows you to avoid the discussion if you are hand-picking the spikes. Okay, so I just put the wire there, record everything, and then do the multi-unit activity. Go home. Okay, another question? Okay. Okay, so with this model, so we have, this is excitatory inhibitory, this is excitatory inhibitory, and we are just so this is going to be our network. That this goes directly to the respiratory activity, or this goes through this loop. And what we want to reproduce is not only the song, but we are just going to be focusing in this pressure activity. Do you remember at the beginning of the talk that I told you that we record also respiratory activity? Well, this is the pressure in the air sac system that we can record while the bird is singing. So the advantage of canaries is that each pressure pattern relates to a specific syllable. So if I can reproduce the pressure, then reproducing the syllables, it's easy too. Okay? So I want to reproduce this part of the behavior. Then I can use the other model, the vocal model, in order to reproduce these other parts. We did that too. Okay, so we have this sparse behavior. So, for example, each color is different neurons. We have like all these pairs, but we want the popular neural activity. Okay, so we do like add all the sparse activity of individual neurons in order to get this population activity, this multi-unit activity. Okay, and what we get is here there are no gaps, but still you have like an oscillatory regime. And the air sac pressure is like this, okay? So these are all recordings, okay? Pressure that we record, neurons that we record, and this is the processing. And if you see that each peak of the neural activity corresponds to one peak of the pressure pattern, okay? Again, the rhythms in the brain could arise from LFPs, or if you sum up all the neurons, okay? And with this, we model. So in the model, remember how was this? This was ER, and then fits to HVC. This ER fits directly to the respiratory, or fits HVC, then array, and then the respiratory. And in here, we want to reproduce the respiratory activity, so the pressure pattern. So ER fits array, and then HVC. So in HVC, we want these specific patterns that corresponds to these specific patterns of neural activity, and then the model is also able to reproduce this respiratory pressure. Okay? So in the model, we reproduce HVC and the syllables. More importantly, in these long whistles, we find neurons at the beginning of one syllable, So this is the pressure pattern. So this is one syllable and this is the other syllable. So it's at the beginning of the syllable, in the middle, and at the end. Okay? And that's the prediction that we have. The prediction is that in the middle, we don't have bursts of phasic neuron. And that is precisely what we are finding in our recordings. And in the modeling, we needed two kicks, if you want, two steps that represent these two. In order, with just these two steps, you can reproduce the whole complex behavior. And what is interesting is that in the modeling, we didn't need this final step. Just with decreasing with array, it's enough to decrease the pressure. We can add it later, but actually doesn't improve what we are interested in, the nonlinear dynamics. So we just leave it without the third kick. So this is, for us, this is just like a security thing, like an overdoing it of the neural activity. What do we learn from this? Because this is a very low-dimensional model. So we learn first that we can generate sophisticated motor activities or complex motor output with simple input. And this is the key of nonlinear dynamics. Simple input, complex output. And we can do it. So how does it work that the brain is not coding for each individual moment in time? It works because that is embedded in a nonlinear system, a neural network that is very complex. So you don't need to be coding in every step of the way. Okay? That's the main message. You just need precise steps, and then the dynamics will take over. Okay? That's important. The other thing that we wanted to test, okay, yeah, so is this. So it's sparse input, complex output. Okay, so sparse input, complex output, and this is specific predictions of HVC activity. Okay, so this is like the slide of the take-home message. This we publish in the journal Chaos. Okay, so we had some modeling, some neurons, and we wanted to put it out in this is a physics journal. So I was talking to a student before, and we can publish either in physics journals or in biology journals or in general journals, but you can have to choose what you want to say about it or what the fights that you want to put out. Another thing that you can say is that this cortical loop through HPC is required to simulate complex song. What does it mean? So this is the cortical loop. If I remove this part, I cannot elicit this. If I remove this part and I just keep this, the only thing that you have is just a response. Okay? Doesn't matter if it's excitatory inhibitor, so it's just a response. So what we are saying is that you need HVC, but you don't need HVC coding all the step of the way. Okay? So that is the modeling. We are so happy. Highlight the importance of characterization behavior when study vocal motor control in songbirds. Rhythms are synchronized with behavior, and a distributed neural code is proposed for the vocal control in the song system. Neural recordings suggest a motor-gesture related neural code. Nobody cares of what we said. Everyone thinks is still a sequence, but we know we are right. And the beauty of science is that at some point people will understand what we are saying, and la historia me absolverá. Okay. No, I mean, that's the thing. I mean, in some years we will really understand more, and we will know who was right, because the important thing is to win.
I have a question.
Yes.
So, is it possible that in zebra finch case it is really rock, but the canary is different?
Yeah, it could be. It could be. I don't think so, because But you're totally right. Actually, we submitted a grant where we wanted to study with Dan Margolis, Todd Troyer, some genetic guys. We wanted to study the difference between species. Say, well, is there a difference in the genetic code? Is there a difference in the way the neural structure is organized? What are the differences between the species? We were not founded. So the money didn't arrive. They didn't think it was interesting enough. But that is a very important question to ask, because we are just generalizing of what we know of one species, but what's the difference between canaries and zebra finches? They're completely right. So they have different behaviors. Are there any difference in the neural architecture, or are there any difference in the genetic things? We know genetics matter for the behavior. It's not that it all is learned. You have genetic constraints. We don't know enough. We need money for that.
Like in the anatomy of the ear of the bird.
Of the?
Are there differences in the anatomy of the ear?
Not that we know. Not that we know of. For what we know is the same chickens that are non-learners or zebra finches, so that we don't know. But still, we are not a huge community. Although we like a lot and we think some birds rock, we are not so many in the world that think that way. So most of the people are studying rats, mice, and all that. So we need more manpower to study more. And auditory, and the auditory region, we know even less than motor control. We need to study more. Yes.
How did you set up the coupling strands between brain regions in your model?
Yeah, so this was it. We didn't spend more time on that. We just wanted to show that it was possible, that just with minimum ingredients, it was enough. So we wanted this. Sparse input, complex output. That's the only thing we wanted to say. Sometimes you have that corresponding one-to-one with one kick, one expiratory pressure pattern, or one kick, dynamics, and something else. And that's the only thing we wanted to show. We were not, because this you cannot measure. So what caring of model, is this a unique, so for example, are these parameters unique, or you have other regions of parameters that can give you the same response? We don't care. We just wanted to show that we wanted to show this, that what we record was similar of what we find in the, what can be reproduced with the model, that these recordings would make sense in a brain. Yeah.
No, well, not me answer the question, but so from this perspective, where is the learning happening in RA? It's not the HB semester here.
Well, I mean, it's in everywhere. It's everywhere. It's everywhere. So to us, and actually the other one that says something like this is Richard Money, and what he said, you have a distributed information. So you need HBC to give you the right kicks, but then you need RA to fill in the gaps. So the learning is in the whole network, not just in HBC. And actually this idea of auditory memories, where are the auditory memories that are what you start? First you have the auditory memories, and then you have the sensory motor learning to acquire the behavior. And the auditory memories are in all the network. So whenever all of these are selective neurons, have selective neurons in these regions. So we think it's distributed in the whole network. Because actually, you also have these mirror neurons. So in HBC, the neurons fighting at the same time when they are hearing or when they are producing the song. And in order to do that, you need some synchronization happening that you don't have a phase lag. We just see synchronization with zero lag. And for that, we think it's a thing of a whole network. But for that, we still need more data and more money. We need money. Okay, awesome. We have 5 minutes for more questions.
Are there a difference between birds that call and birds that fly in the way they sing or interact with each other?
Oh, super interesting. But give me an example. Super interesting. I've never thought about it. There are birds that sing, but for example, tell me a bird that walks and learns how to sing.
Yeah.
Porque the one that learns, we have kiwi, pingüino, emu, the birds that don't fly.
But like the penguin, they kind of like flies in the water.
So no pingüino. So we have kiwi. So bear with me, I'm not a biologist. Birds that don't fly. Kiwi, ostrich, but ostrich don't learn. Then tell me the name in whatever language you have.
To me, there was like a relation with the fusion of the species, like how much I don't know, like something runs in here. Maybe it dies somewhere really far away from him. So you can have like a distribution of.
Yeah, no, it could be, but no, it could be, we can think about it, but we need birds that don't fly and learn how to sing. So ostrich don't learn how to sing, and they have a very simple singings. So ostrich out. Kiwi, no tengo idea. Emu, they vocalize, all of them vocalize, but they don't learn. So that would be interesting. We can think about it later. I think that for now, kiwi, I don't know. But maybe if they don't learn, it's just a correlation, right? So all the birds that don't fly don't learn. I don't know. Okay? Okay. Papa what?
I don't know which one.
Papagayo?
There's
No.
a cartoon who uses this.
Ah, el correcto.
Yeah.
I don't know. Okay, we can do that as a homework to see. But what my yeah. But again, they can communicate and they convey messages without learning, and that could be so they associate different vocalizations to different meanings, and for that they don't need vocal learning. But we should do at least the homework to see which are the species that don't fly, and then to see if those learn. If you just tell me the species, we can do it afterwards. If you tell me what species don't fly, I can tell you if they learn or not. We can do that. We first have to do the homework, because then we cannot do an hypothesis if we haven't done the homework. Okay, guys, do you have a question? Or we just call it a day, okay?
More questions?
More questions? Okay, thank you so much.
Okay, everyone. So Amador will be around for the week. So any more questions, just ask her. But not today, probably. I think she's very tired. So we will restart again at 2, as usual. But it will be here. Okay. And tomorrow is the same. It will be 9 here. Okay. We will. What? Yeah, exactly. Yeah, during the week, so we won't go anymore after weekend. Okay?
Very nice. I like them a lot how they engage.
Yeah, no, they're super
Because
interactive.
I was saying, this is very long. If they don't ask questions, I will die. Because I will sleep myself, you know? like walking is... Yeah.